Thank you for being here. My name is Edoardo Sarti, and I will present iBIS2Analyzer, a new web server for complex coevolution analysis. This work has been done in collaboration with Francesco Oteri and Alessandra Carbone at Sorbonne University, and Francesca Nadalin at EBI. We say that two or more residues in a protein coevolve when they have a propension to mutate together, thus showing correlation in a multiple sequence alignment. By analyzing the coevolution of groups of residues, we can retrieve important information on its structure, function, and interaction. Several methods for finding coevolutionary signals exist, but there still are challenges to be tackled. For example, coevolution signal is easily lost when coming from only one subset of the protein family. Also, the performance of statistics-based methods quickly deteriorates when sequences are highly conserved. iBIS2Analyzer uses the BIS2 method for finding correlation signals in a multiple sequence alignment or a protein family. A coevolution score is associated to each pair of columns, and a square matrix containing all the pairwise scores is thus calculated. Each column in this matrix will thus represent the coevolution propension of one amino acid with respect to each amino acid in the protein. The method thus looks for similar column vectors and clusters them together. Residues belonging to the same cluster are not guaranteed to be structural neighbors, but they will have a direct or indirect, structural or functional interaction. This method is not based on amino acid type frequencies in the input MSA, and is thus robust even when the sequences are very similar, or very few, and do not show much variation. iBIS2, the iterative version of the algorithm, is able to first calculate a phylogenetic tree starting from the input sequences, and then perform such analysis on all subtrees. Thus, coevolution signals present only in a subset of the query sequences will be detected. Let me now introduce you to the web server. iBIS2Analyzer has a simple and immediate interface that allows the user to set several options. First of all, an MSA in FASTA format should be loaded or copied. There are also several pre-computed examples that can be chosen. Once the MSA is loaded, click on the Check MSA button to perform format quality checks, and then set the other options or just go with the default values. Don't forget to write your email address in order to be notified when the job ends. Click on Submit for launching the job, or on View for visualizing pre-computed results, like in this case. The results page not also summarizes in a visual way the outcome of the coevolution analysis, but gives the user the possibility to combine many tools for customizing and refining it. The colored cluster panel collects all clusters that have been found and disposes them along the input MSA. Each integer value on the y-axis corresponds to a cluster. In order to declutter the image, there are multiple selection tools, such as, for example, the selection by p-value, that can be activated by moving the p-value bar or by selecting a specific p-value from the menu. A region of the MSA can be selected through the Zoom panel. Each colored dot in the cluster tells which cluster, position, and subtree it belongs to. The position button selects only the clusters that have at least one hit in the region that's currently visualized. If we dezoom, we can see how these clusters distribute along the whole MSA. We can also select one by one the clusters we want to visualize from the Clusters tab on the right. Clusters can also be selected by subtree. In the Tree panel, click any subtree shown in red, and click on Select on this subtree. Any cluster selection will be reflected on the MSA. The corresponding columns will be highlighted by black frames. It is also possible to only show the multiple sequence alignment relative to one subtree in particular by clicking the corresponding red node and selecting Add to New tab. This is especially helpful when iBIS2 has been run with the option for recalculating the best alignment for each subtree. Results can also be mapped on several structures. By clicking the Structure button from the Results page, the Structure page will open. Here, multiple structures can be uploaded or retrieved from the PDB, and the clusters will be mapped on each of them. The same cluster selection options present in the Results page are available on this page. The user can download all the results and input files. Thank you for your attention.